right? So I'm very excited. But he's going to show, talking about dynamic monographs and using herbaria data and looking forward to the future.
Great. Thanks, Mike, for the introduction, and thanks, Mike and Laura, for inviting me to participate in this colloquium. I'm very excited to be here, listening to all these talks about the use of herbaria. So just as a disclaimer, I'm not going to be showing any genomic study specifically based on herbarium specimens, but I'm going to be talking about why I believe herbaria are critically important in systematics these days, but also for the future. So, because the main focus of this colloquium is on herbarium in systematics and the use of herbaria for in the genomics age, let me start by presenting this paragraph that I really like about the importance of herbaria in systematics that I took from the UC and Jepson herbaria at UC Berkeley. So the first paragraph, the first sentences that I would like to highlight is that what herbaria really are. So herbaria are basically places that hold all our knowledge, all the raw data about biodiversity out there in nature. So they have all the raw information about what species are, what are the characters that differentiate different species, where do species occur, when do they flower, how they differ from other species, how are they related to each other, and so on. So the fundamental unit of information that encapsulates all this raw data is, of course, the herbarium specimen. And I guess that everybody here might be familiar with herbarium specimens because you are in this colloquium. And based on herbarium specimens is where we can score a bunch of data sets, a bunch of data that's encapsulated here. So from herbarium specimens, we can capture a lot of information about phenotypes. And of course, phenotypes are fundamental for all our studies on species delimitations. We understand what species really are. We can score a series of specimens and map that information in our phylogenies to study character evolution and trait evolution. We can study how morphological traits vary in geographic space, studies of what it's called now trait phylogeography, but this is what we systematists have been doing forever. And we are kind of lucky, and it's very exciting to be this time working in herbarium specimens where we have access to high throughput technologies to score phenotypes automatically. There's a bunch of excitement about machine learning algorithms to extract information automatically from herbarium specimens. This is an example on the Solanaceae. This review paper put a couple of months ago, or maybe a year ago, by Toby Kellogg and her colleagues. The herbarium specimens are also the unit where we can extract genetic data, and we have heard already a couple of talks about that, and I'm pretty sure that the rest of the talks today are going to be about using herbarium specimens to extract genetic information. And of course, the genetic information is the fundamental basis for our studies on species delimitations. Again, phylogenetics. If we are lucky enough to score multiple specimens per species, we can do pop genetics, molecular evolution, and so on and so forth. So I'm not going to go in details here because we have a full morning of studies on genomics. Specimens also have really relevant information about our traits, such as phenology. Specimens can be in flower or in fruit, but also they have label information where they have specific dates of flowering time, and we can use that information again for multiple studies. So we can use phenology to inform our studies on species delimitations, of course. We can make inferences about speciation and speciation mechanisms and reproductive isolation. We have heard a couple of talks here already, people doing this, and there has been some excitement lately on using digitized herbarium specimens on studies of climate change and adaptation and responses of plants to climate change, looking at historical phenology. And specimens also have geographic information, of course. All specimens come with a label, and the label has specific data of the locality where the plants were found, where the plants were collected. This information is, for the most part, already digitized as latitude, longitude, and elevation, and for all specimens there have been really big projects for the last 20 years or so on people trying to digitize and georeference all the specimens. And of course, this information, again, is fundamental for all the studies that we as systematists do, particularly in species delimitations, again, understanding how phenotypes and genotypes vary across geographic space. We can study, again, speciation and make inferences about patterns and processes of speciation. And for the last 12 or 15 years, There has been also a big excitement on using specimens and databases of bioclimatic data in particular to map geographic ranges and responses of plants to changes in climate and sort of infer potential changes in expansion of the ranges and movements of species ranges through time. So this is just a series of, I highlighted only 4 types of data that we can extract from herbarium specimens, and this is the raw data that that quote from the Berkeley herbarium is talking about. This is the information that we systematists rely and use for all these studies. And the key is that we as systematists, we conduct all these studies and we synthesize all this information in particular types of works that we do. We write florals and we also write monographs. So this takes me to the second part of the paragraph that I really like, where it says that if herbaria wouldn't exist, florals and monographs would be based on abstractions, because there would not be that raw data that underlies all our knowledge about what species are out there in nature. So I'm going to be talking today a little bit about monographs. I'm not going to be talking about floras. So monographs, for those of you in the audience who may haven't seen, read, or written a monograph, it's this comprehensive work that we systematists write after years of study and analysis. We heard Aaron when he just mentioned this monograph that took 24 years. And this is the place where we systematists synthesize all of our knowledge and our understanding of biodiversity for particular groups, families, or genera, or clades within a genus. And we basically synthesize all these studies that I just mentioned: species delimitations, phylogenetics, biogeography, trait evolution, and so on and so forth. So monographs are this synthetic work where we can provide access to all people to all this raw data that the specimens have. And it's fundamental to write these monographs because not only biologists or systematists have access to this data, but also all people who might be interested in understanding what species occur in a group or in a place. They can access this information and they will be able to understand what the systematists did for deciding what species were, how these plants have evolved, and so on and so forth. So if you imagine the connection between the monograph and the herbarium specimen, basically the monograph represents the end of a pipeline where we start with the specimens, and in each of these specimens we can score a series of data. So we can score phenotypes, we can score genotypes, we can score phenology, geography, so on and so forth. And because this is basically a scoring of specimens, we can create specimen-based databases where for each specimen we have all this information. And what the monograph really represents is just the outlet or the hub that synthesizes this information and presents this information to the world. So this is how I conceptualize monographs. One of the issues, of course, is that specimens and herbaria are not static. So herbaria keep growing, people keep collecting, so more specimens are added to our collections. We can revisit the specimens that we have already studied, we can measure more specimens, we can sequence more genes, we can change information about the locality, we can fix details in our database, but also the database keeps growing. Of course, this feeds into our database, specimen database that it's scored for each specimen keeps growing, and what we hope is that the monographs also reflect these changes, because monographs have to reflect what is going on and what do we know about biodiversity. So one of the issues that we have right now is that monographs end up being these static documents that we publish these books and they are static, but the rest of the pipeline keeps moving, but the monograph keeps static. So what I am proposing is that we should sort of rethink about monographs as these documents that are more dynamic, that sort of reflect our knowledge and our understanding of biodiversity, and they should be updated continuously as more specimens are added to our collections. We should be able to link all this information that comes with the specimen into these monographs and reflect this information in these documents that are continuously updated. And of course, because these monographs are basically an outlet of a database, we should be able to provide the raw data to the audience or to the outlet who is the people who are reading our monographs. They should be able to see where the data comes from, and we should provide access to this data. So what I am sort of proposing now is that we should perhaps think about monographs as this connection between the database here in these little piles and the monograph, and it's connected. And as more specimens are added here at the bottom to our database, or changes happen in the database, automatically this monograph should regenerate every time that more specimens are added or more information comes into the database. But at the same time, the users can query these databases and they should be able to extract information that they want from these databases. And these queries to the monograph should feed back into the database, and the monograph is basically this interface between the user and this database. So what I'm proposing is that monographs should be perhaps conceptualized as just a series of instructions or scripts, if you wish, to run a series of a series of queries to a database, rerun and run more analysis whenever the database changes or the user asks for specific results, regenerate figures, regenerate all the plots, and reformat the text such that we can sort of read it automatically. And of course, provide access to the raw data, dump the database, dump all the information. So I'm gonna try to show a quick demo of something that I have been trying to put. I did it super quickly, it's not thorough, but it's just an idea. But before I move on to show you this, and in case it doesn't work, let me move on into my acknowledgements. So I want to thank Peter Stevens and Toby Kellogg because they have been extremely fundamental in my conceptualization of what monographs should be, and all this work started when I was studying in their labs. Casey Dunn has been providing a lot of feedback on biodiversity informatics, and of course people in my lab at UCLA have given me a lot of ideas and insight on how to do this. I want to thank NSF, of course. This work started a long time ago when I was a grad student. I received support for my dissertation enhancement grant through the international office, and I am happy to announce that I got an NSF 2 weeks ago to to move this work on dynamic monographs. And this is going to be really exciting because there's going to be a lot of workshops and a lot of working groups, and we're going to bring together people from computer science, machine learning, taxonomists, ecologists, and people who are thinking about how to make this work more accessible. So now, let me see if this works. Okay, so I put this very quickly. Recently, I basically wrote an interface in R for what I'm trying to explain here. And what is happening right now is that I have a bunch of R code that is run right now in the cloud. For those of you who are familiar with this is called a Shiny app. This is not necessarily the way to do it, but this is one quick way to sort of get around this. So what's happening right now is that I have a bunch of R code. There's a script that has all the information, and underlying that R code is a database of specimens. Of course, I just took a small sample of a database that I have been working on for a long time. And every time that we load this web page, there's a computer server somewhere that basically launches, starts R, and R runs these scripts and regenerates this text. So for example, things such as, let me see, there's a mouse here. For example, things like these numbers. So these numbers are not written. This is basically R code in the background asking how many specimens are in the database. So if the database grows, of course this number is going to keep growing. But we can do many more things like, such as, for example, these maps, and all these specimens are linked to specimens that actually we can access this information for particular specimens. So this is FC204, and if we go here to the Tropicals database in the Botanical Garden, that's where these specimens are, so we can have access to the actual specimens, information about where they were collected, stuff like that. Some of the really exciting things is that we can start studying patterns of variation and morphological variation in different specimens. So for example, we can have here these plots where I'm plotting longitude, latitude, and the size of the points reflect leaf size. But we can also sort of start inspecting all of these points, and we can start zooming in into particular parts of this morphospace and get better understanding of what's really going on. We can remove some of the species that may be particularly specious and are sort of blurring what we're trying to see here. We can change the axis, and we can do many more things that provide the raw data and access to what we can do, and people can start understanding what variation happens in these specimens. Of course, we can have also other types of plots, such as, for example, PTO length. So all of this is going on in the background. So every time R is running these scripts to regenerate all these figures, and if specimens keep growing, of course, these plots are going to change quickly. We can have also information about phenology. So these are super quick plots on phenology, but again, we can start exploring these phenological plots in more detail, and we can download these figures or download the data. Here you can download the table for phenological information. I have here phylogeny that was inferred also using specimens that I collected that are in the herbarium. I didn't have enough time to link all the tips, but all the tips are linked to measurements and phenological information, so we will be able to link every tip in this phylogeny to all this other extra information, and we can run other analyses, such as, for example, the species delimitation analysis. So quickly, you can run this type of a mixture type of plots, and sort of start seeing the results automatically. And of course, you can also run the same type of analysis with morphology. So these are mixture models that, for those of you who know my work, have been working on this for a long time, and you can use reproductive traits, vegetative traits, or both traits, and you can also have summary information about the results of how many clusters or how many morphological groups you have in these monographs. And of course, you have species, each species will have its own information. Everything is extracted directly from the monograph, and the monograph keeps growing. All this information keeps growing. And of course, you can have access to the raw data, so there's ways to download all the information so that people can analyze this data in different ways. And that's pretty much it. Thanks so much.
Okay, for the time, I'm sure there's probably